Use of antibodies in the isolation and identification of genes and their products. As a first step in isolating the gene that codes for a particular protein, antibodies specific for the protein can be used to isolate the purified protein from cells using affinity chromatography. Small amounts of amino acid sequence can then be obtained from the protein's amino terminal end or from peptide fragments generated by proteolysis. The information in these amino acid sequences is used to construct a set of synthetic oligonucleotides corresponding to the possible DNA sequences, which are then used as probes to isolate the gene encoding the protein from either a library of DNA sequences complementary to messenger RNA or a genomic DNA library. An alternative approach to gene identification uses antibodies to identify the protein product of a gene that has been introduced into a cell that does not normally express it. This technique is most commonly applied to the identification of genes encoding cell surface proteins. A set of cDNA containing expression vectors is first made from a cDNA library prepared from the total messenger RNA from a cell type that does express the protein of interest. The vectors are used to transfect a cell type that does not normally express the protein of interest, and the vector drives expression of the cDNA it contains without integrating into the host cell DNA. Cells expressing the required protein after transfection are then isolated by binding to specific antibodies that detect the presence of the protein on the cell surface. The vector containing the required gene can then be recovered from these cells. The recovered vectors are then introduced into bacterial cells where they replicate rapidly, and these amplified vectors are used in a second round of transfection in mammalian cells. After several cycles of transfection, isolation, and amplification in bacteria, single colonies of bacteria are picked, and the vectors prepared from cultures of each colony are used in a final transfection to identify a cloned vector carrying the cDNA of interest, which is then isolated and characterized. This methodology has been used to isolate many genes encoding cell surface molecules. And that's use of antibodies in the isolation and identification of genes and their products.